Hey Team Bio, welcome to your screencast about the structure of proteins. So today I'm recording this before class. I'm not quite sure how far we're going to get, but I'm going to be referencing some things, partially which you may have done and partially which you may have not done. So we'll see what it feels like. Okay, so today in class, I gave you the model for enzyme action called the induced fit model, where we had an enzyme which had an active site or multiple active sites, and we had this lock and key hypothesis where the substrate fit perfectly into the active site. And it's through the interactions of the substrates with the enzyme that the enzyme is able to catalyze the reaction and lower the energy of the activation through the position of the substrates in the enzymes. Now, this screencast, I'm going to talk to you a little bit about how an enzyme takes on its structure, because it's got a couple different levels of structural organization that give it its very specific shape. Okay, so the first layer of structure within an enzyme is called, or a protein, is called the primary structure. So what I've depicted here is all of these little dots signify amino acids. So amino acids, when they're joined together, which you may have done in class today, but you may not have, they form what's called a peptide bond. And a peptide bond is a covalent bond. And as such, it is very strong. Remember, covalent bonds are strongest types of bonds that can form between atoms. When two amino acids are joined together through a peptide bond, we call this a dipeptide. And when we join a long chain of them together, we call this long chain of 3 or more a polypeptide. Polypeptide. But in order to transform this from a polypeptide into a protein, we need to start to fold it. And the folding takes place starting in kind of the secondary structure. So, if we copy this and paste it down below, and then copy this, paste it below, paste it below. Copy this whole thing and paste. And then copy this whole thing, paste, paste, paste. So we've created here what we call a beta pleated sheet, or just a beta. This is like a B with a long tail at the bottom. A beta sheet. So we've taken a long chain of primary structure, so a bunch of amino acids all joined together covalently, and we've taken that chain and we've folded it into this specific shape, which is called a beta sheet or a beta pleated sheet. Now, I told you before that the individual amino acids are held together by covalent bonds, but the structure of the beta sheet is held together across the rows by hydrogen bonds. So I'm not going to draw hydrogen bonds across this whole way here, but know that these red bonds that I've drawn here are hydrogen bonds. So the same type of bonds that hold water molecules together with each other, so cohesion or adhesion, water to other things, the same type of bonding is happening here between the amino acids that are in their primary structure. So the secondary structure is held together with hydrogen bonds. And remember, hydrogen bonds are not as strong as covalent bonds. And so it means that this structure is not quite as strong as this structure. And it can be broken apart if we apply the right techniques. Okay, the next thing, so that's one example of a secondary structure. Another example of a secondary structure is a word you've probably heard before. And okay, so what I've drawn here is a not fantastic alpha helix. So if you start here, imagine following around these loop-de-loos. So the classic alpha helix structure, kind of like a spiral staircase. So this is another example of a secondary structure motif. And once again, the alpha helix is held together by hydrogen bonding, interactions between the different amino acids that form the linear structure. Okay, so this is another example of a secondary structure. So primary structure held together by covalent peptide bonds. Secondary structures are held together by hydrogen bonds. Okay, tertiary structure is when you take a secondary structure, so a motif like a beta pleated, I want to copy this. A beta pleated sheet, and you mix it up also with another motif like an alpha helix. They would be joined together somewhere. So you might have a beta pleated sheet hanging off of an alpha helix like so. And so a tertiary structure is going to be composed of two or more, usually more, So there are a bunch of different secondary structure motifs, like hundreds, if not thousands of secondary structure motifs. And they're all going to be combined mixed and matched into tertiary structure. And again, these motifs, this tertiary structure, is going to be held in place by hydrogen bonding between the amino acids that compose the linear chain. So once again, holding the tertiary structure in place is hydrogen bonds. And then finally we come to quaternary structure here. And quaternary structure is when we have two or more tertiary structures interacting with one another forming subunits of, this one doesn't want to stay in place, okay, of a larger protein. And once again, this quaternary structure is going to be, these different subunits are held in place by hydrogen bonds. So H bonds hold in place. So the difference between quaternary structure and tertiary structure is composed only of one primary structure. So if we were to start at this end and trace our way all through the protein and get to the end here, we'd only find one chain. Whereas quaternary structure, we're going to start here and trace our way, and we find our way only halfway through the protein here because there are other primary structures that have been folded up into secondary and tertiary structures that are interacting with each other, held in place by hydrogen bonding here. Okay, so why is any of this important? Well, as I said before, covalent bonds are very strong, and hydrogen bonds are not super strong. You can break them relatively easily by interfering with the hydrogen bonding, and that can be done through application of heat or things that disrupt hydrogen bonding. So heat can disrupt a hydrogen bond. A change in the pH can disrupt a hydrogen bond. Cooling can freeze the hydrogen bonds in place. So these are kind of opposite sides of one another. And high salt can also disrupt hydrogen bonds. And as we said before, so this shape that a protein takes on is integral to its function. And so if the shape is disrupted, a process that we call denaturing a protein, it'll turn this quaternary or tertiary structure protein back into a polypeptide, and it can no longer perform its function. And we're going to see that tomorrow in the lab. We're going to apply some things that denature our catalase enzymes, and we're going to see the result on the enzyme activity. Okay, that is all. See you in class tomorrow.